this type of separation of restriction fragments was initially used was in comparing DNA samples from different sources. Restriction sites are not uniform within a species, so individuals have different DNA sequences and therefore different sites and therefore different lengths of restriction fragments that can be compared after digestion and separation, allowing what has become known as DNA fingerprinting or restriction fragment length polymorphism, RFLP analysis. Taking DNA from different individuals, humans, trees, animals, soil, bacteria, any source of DNA, and treating with a restriction enzyme yields a fingerprint that can be compared between individual samples. Sometimes samples are amplified by PCR prior to analysis, but that's done very carefully to avoid contamination. Fragments are separated by gel electrophoresis and analyzed by staining or by transferring onto a membrane and probing with specific nucleotide sequences. The fragments are visualized by fluorescent probes, by radiolabel, targeted to specific sequences, and images are created on film or in electronic format. This is a DNA fingerprint using RFLP analysis. This method quickly became fodder for television crime shows, and indeed this approach remains widely used in the forensic setting. Because it is possible to take DNA from a crime scene or any other setting and compare the fingerprint of the DNA, the pattern of bands, the RFLP pattern, to individuals suspected in the specific crime or to another organism or another species, this comparison allows many applications from identifying paternity to applications in evolutionary biology.